Welcome, Dr. Bohr, to the CERP celebrating 10 years of leading NAD research. CERP stands for the ChromaDex External Research Program. I don't know exactly, perhaps we'll get into this, but certainly you were probably one of the earlier, what we call CERP investigators to be a part of the program. And we've had the pleasure of working with you for a number of years now, and even greater now connection with you because you serve on our scientific advisory board, which is wonderful. We couldn't be more happy. So we'll start out by asking just a couple of questions. We're going to keep this probably to about 10 to 15 minutes, so it's not going to be a lengthy interview, but so please tell us about yourself and your research interests.
Yeah. So I've also enjoyed to work with you, Andrew, and others at ChromaDex from way back. I can't remember how many years ago we started, but it was quite a few years ago, and I've worked with changing leadership at ChromaDex, but always enjoyed working with you for a couple of reasons. One is I strongly believe in NAD supplementation and use NR. And I also feel that the company is very open-minded to the research, following the research. And so there's a good discussion, an open discussion about the results that we get. And so I've always felt comfortable discussing this with the company, and that they were open to things that worked and things that didn't work. And of course, there are no cure-it-alls, snake oils, so nothing is perfect. But I have felt over many years of work that NR comes fairly close to being a really good agent.
Mm-hmm. But along the lines of your research, what have been your research interests?
So my research interests have been in the area of genome instability, DNA damage, DNA repair. I've worked with that for many years. And I have then I worked with that before I entered into first the Cancer Institute, where it's very relevant, and then into the Aging Institute, where it's also very relevant. So DNA damage is really one of the major underlying causes of both cancer and aging. And so that drove me further to look at this process in various aging models, so to speak, and aging in humans and aging in model systems and aging in mice. And that work led us to discover, I would say, a link between the damage and the DNA damage signaling process that then leads to mitochondrial dysfunction. And this goes through depletion of NAD in the cells, and we have found that quite a number of years ago. And so that led us to connect with you and start using NAD supplementation at an early phase, and also many years ago for you. And we have then achieved a lot of positive results in these model systems that relate to both aging and DNA repair deficiency with NAD.
Yeah.
And so for me, it's been kind of two things here. One is that I have felt that we made real good progress in ways to intervene in humans. And I've also felt that we made good progress in understanding some of the basic mechanisms, because we were using NAD to understand the process, the mechanism by which we think mitochondria are defective in neurodegeneration, but also there are a lot of exciting potential and intervention.
Yeah. It seems that DNA repair, or perhaps dysfunction around DNA repair, seems to be a a central hallmark of aging and age-related diseases. And of course, NAD plays an important role in DNA repair. So
yeah, so it has been known to be a central factor in a number of diseases, and also in aging itself, where DNA repair was shown to decline gradually. But then I would say that then other theories come up and other theories go down. It's all
sure.
a bit of a cyclic thing, but this idea that the DNA damage and lack of DNA repair is important has persisted for quite a long time. And sometimes it gets strengthened, sometimes it gets weakened, but it's there. And a lot of people believe it's important.
Mm-hmm.
And so this kind of leads me to say that when we intervene within because of that mechanism, then, as you and I often has discussed, we wanted to target people who have DNA repair deficiency or lower NAD.
Right.
They are the ones that would benefit the most from this intervention.
Right. So actually, that leads me to my next question. If you could describe some of the many publications that you've been a part of now that involved researching Niagen, which is ChromaDex's patented nicotinamide riboside, or NR. You just referenced it. If you could describe some of those studies, and there are many, of course, as we know, but why you think this research is impactful.
So one of my interests in approaching, well, aging is a very broad field.
Sure.
It covers so many things, and we're all affected by it at so many levels. And so early on, when I started my department at NIA, we were looking for the best approaches we could take to understand aging. And an approach would have been to look at very old people, for example, and see how they were doing so well of the centenarians. But another approach that was closer to understanding mechanism was to look at these individuals, these diseases where the patients appear much older than they actually are. These are called progerious or premature aging disorders. And here we have a list of those, just a handful of diseases where it's been markedly found by clinicians over a long time that they look so much like early aging. And they are monogenic diseases. They're diseases with mutations in certain genes that then underlie the mechanism. So by studying those diseases, we would look at a condition that resembled normal aging. It's not exactly normal aging, but it resembles normal aging. And we would have an underlying understanding of the mechanism leading to that. So combining the mechanism, understanding what that gene does in the system, in our organisms, together with an intervention would then be a very good way to go. And that's what we decided to do. And then over the years, we've looked at a number of these conditions. And for example, to be specific, as when you're asking, is a condition called Cockayne syndrome that has always interested me a lot from the viewpoint of the patients that I find very compelling to help for a number of reasons I could discuss with you, and from the reason of mechanism, because the genes and deficient in these diseases play very important roles in DNA repair. And one of my earliest contributions to work in DNA repair was to help understand what the role these genes play, what are the roles these genes play in DNA repair. So for me, it's been a really long and interesting story to continue working with that condition, from understanding the mechanisms of what the protein that's coded for by the gene, what that does, to try and help these patients by intervention. Mm-hmm. So that's an example of a rare disease that's very important for very severely affects the individuals for neurodegeneration, where we don't have a treatment at all. And I feel that by understanding such a disease, although it is a rare disease, it gives us great insight into the aging process itself, because it in so many ways resembles the aging process.
Right. Well, thank you for that. I guess probably you could describe also here from your perspective is, why is NAD so important to health based on research that you've done?
Well, I mean, there are many things that are important to health, but NAD is essential and participates in so many processes. And it just happens that the processes that I'm interested in, that I think are important in aging, involve NAD either as a substrate for an enzymatic reaction,
right,
or as a co-functional thing. So I think that was of course, not my idea. People have studied NAD for a long time and came up with that many years ago. I've just written a big book chapter of some of the history of NAD. And then so I think that many people subscribe to the idea that that's a very important component, but by no means the only important component understanding aging.
You know, I don't want to digress too much, but you know, my background is in nutrition.
Yeah.
And the nutrition community sees NAD as a way to prevent pellagra.
Yeah.
The deficiency disease.
Yes.
But this whole idea that NAD may be involved in the aging process or may affect the progress, the susceptibility to chronic conditions, degenerative conditions, this is not something, for some reason, the nutrition community, research community has really paid a whole lot of attention to. And it kind of surprises me.
Yeah. It is surprising, because that's how it all started with pellagra,
right?
You know, and so one would think that they would pursue it, but maybe it's just a matter of time before they realize that.
I hope so. Well, certainly the NIH is getting more and more interested in this area, as you know. The NIA, National Institutes on Aging, where you spent many decades working, has started to get much more interested in NAD, as you know. So the next question I have for you is, what advice might you have for junior scientists who are just getting into this field of research around NAD precursors? What advice might you have for a new young investigator?
This is what I'm confronted with a lot. I give talks all the time, and yesterday I gave a talk here at Berkeley where many of the young people asked me that question. What could I do in this field?
Right.
Exactly that question. And you know, some people say, well, NR, NAD, these things have been around for a long time. We know everything we need to know. And then at the same time, I pointed out that there are so many open questions to address. And I think by studying this field, you have a wonderful opportunity to study anything from biochemical interactions to cellular interactions to mouse models to human clinical studies. And there's so much to learn about the role of NAD at the mechanistic level, and there's so much to learn still about therapy and advice as to who should take NR, for example. And I think it's a really important question right now, for example, at the clinical end of things. Where should we really use this? What are the most important clinical studies to do now? Why are the limited positive results under some circumstances? There are really big challenges. And I think I point out to them that I think the safety is very good about NR. So compared to many other things that they could work on, that are being used for intervention in aging, this has the big plus that it is safe compared to, people ask me a lot about rapamycin and other things. They're also hopeful, but they're not so safe.
Right.
So we want to work with something, I say to them, that we can tell people to safely take for years to come, because it's not going to be one day here with something very volatile like NAR is.
Right. That's excellent advice. And so for you, of course, we at ChromaDex, myself, my predecessors have worked with you for a number of years now. How has it been being an investigator in the ChromaDex External Research Program? How helpful has it been for your research?
Yeah, and I think your next question has to do with also what could CERP do more, so to speak, so I could take them under one. I would say
sure.
That I have really, that's one of the things I like about ChromaDex, is that you have that program. And I think it's great to coordinate with you and Yasmin and others and get good advice and good help. And I think it's really positive that the company supports so many studies around the world, and that they do that, again, with accepting whatever data comes in, pretty much, not trying to sort out things that could be negative. And we work with negative things, and I've seen that, and luckily have not been too many negative things, but we're open to it. We're definitely open to things. There are a lot of issues around cancer, for example. We really want to remain very open to that, and I know you do, and that's one of the things that I enjoy. I think it's a great idea with CERP. I think it really is, and at meetings we see all these things that are going on. And Yasmin would present at the FACET meeting, for example, a poster. We all see this, and it's very exciting to hear. I think that maybe even more could be done to connect the investigators, because you could make some connections between people. They could have, I mean, it would be wonderful to have a CERP meeting and bring everybody into a nice place and have a week's meeting and discuss with everyone. That would be fantastic. Now that's maybe not realistic, but one could maybe have some focused Zoom meetings between different investigators, because I think a lot of us could learn a lot from many of the others, and we don't really regularly see them, and unless we get informed, we may not be aware of what others are doing. So CERP could be a great connector. I know that there are, websites that are shared that maybe could be things like that. But I think to have more direct sort of associations and say, maybe you talk, hi, why don't you talk to this person? We could have a Zoom or something that, and you could then participate in the Zoom. I guess some of you, that'll take a lot of your time, but learn more and connect people more, I think would be great. But I like the program, and I'm very pleased to be one of your selected interviews for this, because I totally support it.
Wonderful. I just want a quick follow-up question to that is, in your many years of experience researching maybe other substances, other compounds, have you come across a program like CERP, or how unique has this been from your perspective as a researcher?
Yeah, I can't readily think of having come across this, or although if I think back about things that we were interested in. For example, I was just discussing with some of the students that in my time in DNA repair, we used to have complete sessions on the effect of green tea, for example. And at that time, we thought that green tea was a wonderful substance, and it shows stimulation of DNA repair. And there were meetings around that, and we met all the people that were doing that. It kind of has waned. I don't know. I still think green tea is good, but for example, we had that. I think that CERP is unique, but even more could be done to bring things together. And I also think that if there is any way to convene a meeting,
Mm-hmm.
I mean, I know it's a funding issue, but I think that that's a really useful thing to bring people together and talk together and to connect, and not just by Zoom. But I know that's a big thing to ask, but I think that would be really cool. But I think it is unique. And again, I am also happy to help with anything that I can do to you and Yasmin. And I think it's really a great program.
Great. Well, those are the questions that we had for you, Dr. Bohr. We, of course, appreciate your many years of contribution to the field, to NAD research, helping to raise NAD awareness, and look forward to continuing to work together and continue to move this field forward. So thank you very much for your time.
Thank you for having me, Andrew and Yasmin.